Good evening, friends. I'm Dr. Banerjee here from Jabalpur. This is my for Ideal Genomics Next Generation Sequencing Lab. I will take you to a tour to my genetic lab, and you can see this genetic lab as a. If you come to this area, this is a molecular pathology lab. And this molecular pathology lab and PCR lab is basically meant for molecular pathology. And this is very important for why I am taking this lecture from my lab, because genetics and molecular genetics and infectious genetics, these are so important nowadays that the diagnosis becomes very precise and easy if we apply genetics in our day-to-day practice, be as a clinician, be the MD medicine, be as MD pediatrics or super specialties or gynecologist and especially pediatricians. This area of PCR lab is the molecular pathology lab. If you can see here, this is the donning and doffing area for infectious pathology. And this area is for extraction of DNA from the infectious material like chikungunya, like COVID, like malaria, like any pathological bacteria, virus. And this is so important for those practitioners who are making a diagnosis, and especially in ICU patients where you need a specific antibiotic, you need antibiotic resistance, this detail also, which bacteria is developing resistance and which is used. Second, if there is a fever, you don't know the reason, then there are many we applied genetics for the clinicians. Why we should know about our study in genetics? Why we should study for our Huh? But it is okay. Yes. Now I think people can listen to me and how to share this slide. मैं भी दिखाऊं. Why we should know or study genetics? That is very important. For pediatrician, the congenital birth defects. For congenital birth defects, for dysmorphism, for developmental disabilities, for neurological and neuromuscular disorders. आवाज नहीं आ रही. Am I audible for my participants? Hello.
यस सर, आवाज आ रही है सर, आवाज आ रही है सर.
Thank you. Actually, मेरी आवाज मुझे भी सुनाई. Why we should know or study genetics? For pediatricians, it is congenital birth defects, dysmorphism, developmental disabilities, neurological and neuromuscular disorders, metabolic disorders, inherited hemoglobinopathies. बोलने का मकसद ये है कि जो pediatricians हैं, those who are looking after the neonates and young adults, even adolescents, sometimes there are congenital birth defects and dysmorphism. There are developmental disabilities, neurological and neuromuscular disorders, very difficult to diagnose sometimes. And at that time, you need a genetic diagnosis. Now, how to do that genetic diagnosis and how to go for genetic diagnosis, then we have to have some knowledge about the applied genetics in our day-to-day practice. When I say about the genetics, it is not only the gene itself, it is the chromosome, it is the whole genome, whole genome, and it is all about the all DNA structure in our body. If we can identify any abnormality into that, then we can go a pinpoint diagnosis. And very importantly, whether this will happen again or not, that is also very important. Now, in obstetrics and gynecologist, why I am doing this test in my lab, why I put this lab, this is so important for having a healthy baby, diagnosis the baby with a disorder in utero, and even because I am doing in vitro fertilization also. So for pre-implantation diagnosis of many de novo and inherited disorders. Many patients who are coming to me with a diseased baby earlier and now they want another baby and they are so fearful about this thalassemia, sickle cell or Duchenne muscular dystrophy or spinal muscular atrophy that the baby if they are having in their house, with them is such a devastating thing that they want that another child, if this type of happens, then it will be a devastation. So it is very important to understand why it happened. Can it be prevented? Can it be diagnosed? So these are so important applied application of genetics that a clinician can understand little bit of genetics, then his knowledge and his application of knowledge goes very high and the pinpoint diagnosis becomes very important. In the same way, the physicians, they can do a precision medicine. They can individualize the drug therapy. Whenever they are managing any fever of unknown origin, then what is the reason behind that? Is there any virus working inside there? In physicians, like in respiratory panel, we do multiple panel of viruses in a single go. We can do pinpoint diagnosis about the pathology so that there should be a correct drug treatment. There should not be any wide spectrum, like we are giving wide spectrum antibiotics in a very high amount of antibiotic of a very high grade, which is not at all required in multiple of times in ICU. For the pathologist, one of the very most important group of this talk is that they are in the field of actual diagnosis. And if my pathologist friends, they apply genetics into their day-to-day practice for diagnosing the infectious disease, it takes many days to do culture and sensitivity. They are not able to do the even culture also. So what is the diagnosis? Diagnosis doesn't come. So if we can have pinpoint diagnosis of the infectious agent, how it can be done? It can be done until unless we get conversed, we get little bit friendly with the genetics. Because genetics word is so, sometimes we don't want to go into that because we never, हमको कॉलेज में पढ़ाया नहीं गया ठीक से, हमको कोई डायग्नोस्टिक जेनेटिक्स करके नहीं पढ़ाया गया. और जब भी आप अभी भी जितने भी आप डायग्नोसिस का देखेंगे, जितने भी आप अपने ट्रीटमेंट में देखेंगे, ओपीडी में देखेंगे, ऑपरेटिव वर्क में देखेंगे, एवरीवेयर यू ओपन द बुक एंड इट स्टार्ट विद द application of genetics. So we are, although we have not been read in our undergraduate or postgraduate, if we can have this knowledge, then we can apply this knowledge. And in oncology also, the therapy is now decided by genetics. So I am going into with this इंट्रोडक्शन. मैं बहुत ज्यादा डिटेल में नहीं ले जाने वाला हूं, बट आप उसको कैसे यूज करेंगे वो बताने वाला हूं. दैट इज द ओनली माई एजेंडा. इफ दिस टॉक इज टेकन वेल बाय माई listeners, then we can go a little bit into deep. But I am not going into the deep. मैं केवल अप्लाइड में रहूंगा और उस एप्लीकेशन में आप कैसे एक नेक्स्ट जनरेशन सीक्वेंसिंग लैब, माइक्रो एरे लैब, कैरियोटाइप को कैसे यूज कर सकते हैं, उससे आपको क्या फायदा हो सकता है. माई पर्पज ऑफ दिस टॉक इज ओनली दैट. I don't want to teach you genetics. I don't want to make you that you should know everything. But if your inclination towards genetic is there, then your diagnosis, your precision, your treatment becomes more specific and it will take less time to treat the patient. So genetics in nutshell. If you see, this is a cell, multiple cell. We are as an organism, we have tissue, and with the tissue we make organs, and with the multiple organs we become a individual. So these are the cells which are either of liver, of kidney, or brain, or any organ, you can say any tissue. But this is made of unit of by cell. So this cell is a unit of our whole body, and this cell contains another round substance which is called nucleus. If you see this nucleus, then in this nucleus there are two threads of a chemical which is called deoxyribose nucleic acid, which is coming one from my mother and another is coming from my father. Each string of a chemical which is 1 meter in length, one from my mother and one from my father. And this is common with each and every cell of your body or our body. If there is problem in the writings of this thread, then there is a problem. This thread is 2 meter long, and whenever our any organ or tissue which proliferates, like in our intestine, like in our in pregnancy, like any tissue which is proliferating, that means we are making cells from one cell to second cell, then two cell to four cell. How this happens? That means these threads has to replicate in each and every cell, one from father and one from mother. That means each and every thread has to be photocopied or get copied by any means so that one go from here to there and another go here and this. I'm not making the color specific now, but this has to be replicated or duplicated individually. That means the red thread becomes another red thread and green thread becomes another green thread, and each of the thread will go into the individual cell. So another cell with the same type of writings in the thread becomes the copy of the cell. If the same copy is happening, then this is okay. That means thread is going into that and same writing. But if this writing becomes abnormal in the progeny of the cell, then this cell will not be same. And you very well know, if the parent cell, the progeny cell, the daughter cell is not like the parental cell, then this is not good. This is called anaplasia. Otherwise, it is hyperplasia. If there is anaplasia, then there will be formation of abnormal cell growth that is called cancer. To understand all these things, we have to know what is happening with this thread. Now, if this thread is being replicated or copied, this cell division takes place within 4 to 5 hours or 7 hours or 8 hours. And this is 2-meter thread. And if we make a copy from one side to other, it will take days. So what nature does, it breaks this thread into multiple pieces, and each piece is given to a shop, photocopy shop, so that they can make their own, and then you can stitch this thread, and this thread is made again. Like a book, if it's a 4,000-page book and you want to photocopy is 1 hour, then one photocopy shop will take it for 3-4 days, but you have to return the book in 2 hours. Then what you do? You take out the chapters separately and you give that chapters to us individual shop. And when they give within 1 hour, all book is within 1 hour and get copied. You stitch them, give the parent book to the library, and you will get the copied book. In the same way, this thread also does this. In how many pieces? It makes it in 23 pieces, each thread. That means each thread will be, I'm making it in red so that it is easy to see. Each thread is made pieces into like a chapters of the book. How many pieces? 23 pieces. That means every piece is having some part of the DNA piece. This 23 piece of each and every thread is called 23 chromosomes. So one thread is making 23 pieces, another thread is making 23 pieces, and they are making their photocopy. So at one time, the cell will have 23 pieces from maternal thread, 23 pieces from the paternal thread. So there will be 23 into 2, that is 46. So each euploid or normal cell will have 46 pieces of this thread. But normally, when cell is not dividing, then this book is in a bound manner, in a thread like. You cannot see whether it is chapter number 1, 2, 3, 4, 5, like 23 piece, 23 chapters. The whole book will be seen as a one book. But when the cell divides, then these chapters get divided, separated in 23 from paternal, 23 from maternal. So you can see 23 into 2 pieces in a cell. This is called metaphase. I don't go into that detail, but in metaphase, we can see this chapter separately. When in a book, all chapters are of not same breadth or same width. In the same way, in the chromosome also, they are not of same size. Someone is large, someone is small. So when we arrange these chapters in size wise, this arrangement is called karyotype. So if any cell, which is a daughter cell, is not having 46 chapters, then we can say that this cell is having abnormal chapter numbers, abnormal chromosome numbers. If this is a somatic cell and happening like this, then the next cell will be 47 or 48. Then it is anaplasia and it is a tumor. So you can do a karyotype of the tumor also and counting the threads, counting the chromosomes. In the same way, in Down syndrome, the extra chapter is number 21. So normally there should be 121 here, 121 here. But an extra 21 if comes, then it is a 321 cases. Chromosomes are called, any body is called som. Som means soma. Soma means body. Chroma means color. So colored, when you stain them, they take color. So they are called chromo, color, som, body. And mostly all of our cell will have number 2 chapter from my father, number 2 chapter from my mother. So each and every chapter will have 2 copies in a normal cell. So it is a disomy. If one chapter is missing, then it will be a monosomy for the the particular chapter. If it is 3 chapters, then it is a trisomy for the particular chapter. Like in Down syndrome, 21 into it becomes 3. In Edwards syndrome, 18 number chapter become 3. In Patahus syndrome, it is 3. These are very important in prenatal diagnosis. But sometimes these embryos, these fetus, they take birth and they go to the pediatrician. Then pediatrician has to make a diagnosis, what is the problem. So it is a chromosomal problem, numerical defect. Now, if I open this cell will have chromosome, which are called the chapters. Now, if I open the chromosome, then what we will see inside? If I open this chromosome, and if you see this chromosome, there are 2 threads. One is this, and what is this? This is the copy of this thread. Because one has to go here, one has to go here. So it has made its own copy. So it might be my maternal 21 chromosome. So it makes a copy of its own. In the same way, there will be a copy of my paternal 21 chromosome. It will also 2 copies. One copy goes here, one copy goes here. In the same manner, all the chromosome has to be duplicated so that it can send its copy to the progeny cells in a same number. Whether it was 46 in a parent cell, it will become 46 in the daughter cell also. Now, I have told you that these are the threads which are 1 to 23 in, but actually they are a single thread. But at the time of division, they become separate, so I can count them. So 1 to 23 from parent 1, parent 1 to 23 from parent 2, that is mother and father. So that means this is actually this thread which is taking a shape of a chapter. So this is a chromosome. Now, if I open this chromosome, then you can see this chromosome and inside there will be the part of the DNA thread which is carrying. So these are all packets of DNA which are packed into a sac, and there are 23 sacs normally when its cell is dividing and it is visible. Otherwise, it is not visible. That means each and every chromosome is having part of the DNA either from the parent, either from the father or mother. That means every chromosome is having part of the DNA. Now, if I see through microscope, I can count the chromosome like this. This chromosome can be counted 1 to 23, and 1 to 23 total should be 46. I can count. But if there is a problem inside the chromosome, it is missing. Then when I count the chromosome, the number will be 46. But actually part of the DNA inside the chromosome, either 1 or 2 or 3 or any chromosome, suppose this is deleted or missed, which is not visible by microscope, then you will say the karyotype is 46, baby is okay. But baby is having disease, so it is not a chromosomal disease, chromosome is okay. So why? So I have to open the chromosome. When I open the chromosome, I can see there are this DNA ki ladi. That means this DNA contains some genes which are actually missing from this baby, which is creating some protein. And mind it, most proteins are enzymes. And if enzymes are abnormal, then there will be problem in the metabolism. If there is a problem in metabolism of any individual, there will be disease. Or it creates some abnormal protein which creates some precipitate inside the cell, then it will create problem. If there is a problem with the protein which makes the structure of the body like hemo, or makes some the globin chain in hemoglobin, or lipoprotein, low lipo LDL receptors, or insulin receptors. These are all derived from a genetic gene activity inside the chromosome because of some active part of this ladi, active part of this thread, which is important to make for that protein. I'm coming into that detail. If this part is missing, you can see there are 46 chromosomes, but there is disease. That means there are some problem inside the chromosome which is not visible by microscope. So they are called submicroscopic deletion. If it is doubled, which is again the chromosome number will be same, then it is called submicroscopic duplication. So these diseases, which are because of submicroscopic or intrachromosomal deletion or duplication, will not be visible by microscope. Then how you diagnose them? So there are methods. I'm coming into that also. Mind it, nature has given us every chapter, one from father, one from mother. Even every page of that chapter equivalent from father, equivalent to that from mother. That means every writing mostly are 2 in number. So there are 2 copies of each letter written inside one thread of a maternal derived thread or paternal derived thread. That means each and every part is in 2 copies. And nature makes balance from these 2 copies so that we work normally. Now, if this part of the chromosome which is one derived from my father, and there will be another chromosome which is normal from my mother. If my father supplies a chromosome which is intrachromosomal defect, intrachromosomal defect, then this part will be missing and there will be a difference in the copy. So submicroscopic deletion and duplication are also called copy number variation. These copy number variations, if they are very important for any gene, anyway, suppose it makes a very important protein and you need equal amount of protein produced by both the party, one from the mother, one from the father, and one is missing, then there will be imbalance. And this part of the chromosome is big chunk of DNA. So you can see many genes will be missing or duplicated. Then how to diagnose submicroscopic deletion and duplication or copy number variation, which is not possible by microscope. So you need another method, which is called microarray. How it takes place, I will go into detail in my second part of the lecture. I'm not going into this lecture because these are the method of diagnosis of chromosomal defects, submicroscopic chromosomal defects, which are copy number variations. Why we study copy number variation? Because there are imbalances in multiple of diseases which are not being able to get diagnosed because lack of knowledge of genetics, and we say it is लाइलाज है, treatment नहीं हो सकता. And the same thing, father again sent the same chromosome, another baby takes place. Or the baby is having some problem and we cannot treat it and we cannot do any justice to them. So we have to know about this submicroscopic deletion and duplication, which are much common in our population than chromosomal defects. Because people can have life with them, but with a defect in them in problem. So this is called microscopic chromosomal defects, submicroscopic chromosomal defects. Submicroscopic chromosomal defects are chromosome is there, which is visible in light microscope. But, and very important thing, another misconception is that how to see the chromosome. Chromosome are very well seen in high power microscope. You can make oil immersion and very well you can take a photograph. But inside the chromosome, if it is a very small part which is not visible through microscope, is called submicroscopic deletion and duplication, which is much more common. Third, if we open this chromosome, this thread is coming here. This thread, when we again open this thread and see what is happening in the thread, you can see this thread, which is 1 meter long of each parent, and this thread is going. And when you magnify this thread is like this. So what is this? This thread is called, if you see, this is the whole thread. And when there is a division, there are multiple pieces of thread. How many pieces? 23 pieces. One from father, another from mother. So there are 23 pairs. These are called chromosome. Inside the chromosome, if there is a deletion or duplication, these are called submicroscopic deletion and duplication. Now, if I open this thread will look like two threads. This thread, when we look into that, then there are multiple writing over here. These writing has only 4 characters. Like in Hindi, in English, how many characters? There are 26 letters, no? English, 26 letters. And with this 26 letter, we make dictionary. That means we make words. Word means we understand something from that. If we say go, then you go. Come, you can come. In the same way, the genetic is with 4 letters. चुप करने को थोड़ा बाहर जा। A, T, C and G. A, T, C and G. These makes a different combination and permutation so that it makes words, and these words are written over here. Each and every word which is being translated and actionable is called gene. Now there are multiple places where these letters are there which is not actionable. So these are called junk DNA. So this is a genetic DNA. This is a gene which is actionable, and this is not actionable. So if I make an analogy with this, analogy with suppose you are walking in my city from one area to one end to another end, there will be a road. When you walk on the road, there might be an airport, there will be a hotel, there will might be a school, there might be some defense staff, there might be university, there might be another school. So when you walk on the road, then there are part of the road where there are something which is important that speaks itself. That airport, it allows the aircraft and everything. If it is a defense establishment, there will be a defense area and all work. If it is university, it works in university. So these area are actionable part of the thread. These are called gene. That in between the road, in between the genes, there are some part of the road which are not having any establishment, but it helps you to reach over there. So at present, they are called junk DNA, but it is not junk. It is not actionable at present, but it is very much used in our DNA fingerprinting business. So I'm not going into that detail. So there are many genes on a thread, and this thread is packed in a sac, which is called chromosome. So every chromosome is having its own actionable part that is called gene. Any part of the chromosome, if it is missing, then many genes are missed together. So it is a great loss, although the whole body is visible. So chromosomally, it is okay, but look wise, it is okay in karyotype, but the airport is missing or defense establishment missing. And this is a very important thing. If it is missing, then whole city or even the nation might be in trouble. So this is called submicroscopy. But if you see this part, then this is the individual part of this thread, which is working as an, suppose, airport. So if I make an analogy of if I make an analogy of my airport, then this is the gate. You enter into the gate. There is a this taxi stand. Then you go into the app. When we go inside the airport and check in and all, there is a runway. And when plane comes out, there is an exit. And again, it comes out through exit. So this is an airport where a gene is called. Now, within the gene, this area is important. That is taxi stand. This area is important. That is Agaman. That is aim. Passengers are coming and they will go. This is the runway. And this is the exit. And this is again the exit. That means within the gene, there are many areas which speaks itself very important. That means within the gene, there are part of the thread which is not used, but it connects them together. So this gene of airport is having part within the gene which is not usually working. this part which is working is called exon. And this part which is not working is called intron. This is a very crude way of understanding, but for the making of understanding, you can make it out. It is an exon, it is an intron. So in a gene, there will be multiple exon, multiple intron. Whenever this airport works, this intron is not required. They should be collaborated together. यदि कोई मिसक्रिएंट घुसता है तो उसको ये जगह ये जगह ये जगह ये जगह ये जगह ढूंढना पड़ेगा. That means these are very important things. So these exons are the main area of working which makes messenger RNA and it makes protein or enzyme. अब ऐसे ही पूरे थ्रेड में बहुत सारे जीन होंगे. दिस जीन में बहुत सारे एक्सॉन होंगे. इन सब एक्सॉन को यदि मैं एक्शनेबल एरिया को एक साथ कर दूं तो हम उसको ग्रुप ऑफ एक्सॉन बोलेंगे. ग्रुप ऑफ एक्सॉन को एक्सोम बोलते हैं. तो हमें यदि कोई डिजीज डायग्नोज करना है जिसको हमको पता नहीं है, तो डिजीज होती है प्रोटीन के डिफेक्ट के कारण. तो हम पूरा थ्रेड का पूरा रास्ता पहले क्यों खोजें? हम एक्सॉन्स को आइडेंटिफाई करें जितने भी एक्शनेबल एरिया है उस मैटरनल थ्रेड में या पैटर्नल थ्रेड में. If we can do this all exons together, then it is called exome analysis. Now, if we get a defective gene or abnormal person in this area, then we can identify this is the culprit which is doing this disease. And we can identify the amount of protein it is being produced by that. If it is less, then we can give this protein extra. Or if it is an abnormal protein, then we can target this protein production by interfering this mRNA. So we can diagnose and we can treat even by knowing the exact location. Now, if we don't get any information in exome sequencing, then what we do? हम फिर पूरे थ्रेड को देखते हैं. पूरे थ्रेड को देखते हैं. इस पूरे थ्रेड में हम एक एक अक्षर देखते हैं. यदि पूरे थ्रेड का एक एक अक्षर देखेंगे टाइम लगेगा. इसको क्या बोलते हैं? होल जीनोम सीक्वेंसिंग. तो होल जीनोम सीक्वेंसिंग, एक्सोम सीक्वेंसिंग, माइक्रोएरे and chromosomal analysis. These are the methods I want to discuss today. How they are used for disease diagnosis, that will be in our next talk. And I'm going, there are aberrations, there might be mistake in the code. So levels of mistake might be a chromosomal, that is numeric or structural, submicroscopic, which is not visible through microscope. Mutation means there is a problem. Mutation comes from mutiny, that is there withdraw, there is disturbance. So there might be a point mutation, there might be a deletion of a letter, there might be multiple letters written together which create nonsense. There might be shifting from one chromosome, like number 1 chromosome here and number 21 chromosome here. If part of this chromosome comes here and part of the chromosome goes here, then it is a balanced translocation. Total number of DNA will be same. But the breakpoint, what happens here and here, if carries an important gene, then it might create a problem with the next progeny cells that might create a tumor. So Philadelphia chromosome, you have heard about how it happens. Though in oncology, if I identify this type of arrangement, then it helps a lot in diagnosis. It helps a lot in treating leukemias. It helps a lot in lymphoblastic leukemia, plasma cell tumor, and whatnot. Even with this, the treatment can be decided. So you can understand in breast cancer, in any sort of this cancers and all, osteosarcoma or anything, the treatment becomes more specific. You can create an monoclonal antibody against this protein, which are end by AB, that is rituximab, whatever it is. There are multiple monoclonal antibody which acts on this abnormal protein and it can neutralize this abnormal protein. So patient can be treated specifically, but you have to know about the genetics of the tumor. There might be a missense or nonsense mutation. It creates a change in this letter writing, which suppose A is changed by T, adenine is changed by thymine. So something abnormal protein might happen, or the protein synthesis might stop. So these are useless nonsense can create disturbance. There might be frame shift. I'm not going into that detail. So the limitation of mistake within the intron. This is the airport. There is this area of taxi stand. Incoming of the passengers, runway, outcoming passengers, exit. If this is happening inside these are called exons. There is a problem in the airport. But if it happens here, then usually यदि यहाँ का रास्ता टूट भी जाए थोड़ा बहुत, हम गाड़ी निकाल के ले जाते हैं. तो within the intron, not much problem. But within the axon, अभी यहाँ का एप्रन का कैनोपी टूट गया, अब आदमी के ऊपर गिर गया, तो दैट इज अ प्रॉब्लम. सो इट इज बैड. विद इन रेगुलेटरी, अब इस एयरपोर्ट को भी रेगुलेटर करने के लिए एयरपोर्ट अथॉरिटी ऑफ इंडिया है. इसका ऑफिस इसके बाहर कहीं बैठा है. इसको यहां से रेगुलेट करना है. यदि यहां पर प्रॉब्लम आ गया, आदमी नहीं आए, या फिर कुछ ऐसा दिक्कत हो गया कि यहाँ पे लोग काम करने को राजी ना हो, तो क्या एयरपोर्ट चलेगा? नहीं चलेगा. सो दिस इज अ रेगुलेटरी एरिया. दिस हैपेंस विद ऑल फंक्शनिंग यूनिट्स. सो इफ इट इज अ रेगुलेटरी एरिया, डेंजरस. सो इन पैथोजेनिक प्रॉब्लम करने वाला चेंज, अब कैट मन से बिल्ली, अब उसकी जगह कट लिख दिया, मतलब काट दो, एकदम ही अबनॉर्मल हो गया. लॉस्ट मैन और लास्ट मैन यदि लिखा तो चलेगा, कोई ज्यादा फर्क नहीं पड़ेगा. बट देर इज अ पैथोजेनिक चेंज विच माइट क्रिएट सम प्रॉब्लम, बट इट इज नॉट अ वेरी डेंजरस थिंग. अनसर्टेन सिग्निफिकेंस, कलर का स्पेलिंग ब्रिटिश और कलर का स्पेलिंग अमेरिकन, नो प्रॉब्लम. सो देर आर चेंजेस विच आर inside the body with single letter change in the gene which create either a pathologic protein, that means it creates absence of enzyme. अब बहुत सारे डिजीज जो मेडिसिन में होते हैं, लो डेंसिटी लाइपोप्रोटीन के रिसेप्टर का हो गया, इंसुलिन का हो गया, ग्रोथ हार्मोन हो गया, एनी टेक एनीथिंग, ये all acts through ligand and their receptors. If there is a problem in the receptor, it will not go inside. If there is a problem, receptor is a protein are found by DNA. So any change which creates a disturbance in the protein production or abnormal protein will create a disturbance in the body function. So we have to identify the level of mistake might be chromosomal, numerical, or it may be extra chromosome or missing chromosome. It might be translocation like BCR, AVL, and Robertsonian translocation. There might be aberration within the sequence, that is sickle cell anemia, Duchenne muscular dystrophy, Fragile X. I'm not going into this uniparental disomy. I'm not going into mosaicism at present, but there are method of inheritance. This will be in a later part of my lecture. So what tools to use? Upcoming topics of the day is level of mistake, chromosome, numerical can be seen by cytogenetic technique. We do it regularly. Very easy to do with lymphocytes. We do karyotype. We can do fluorescence in situ hybridization. No information about submicroscopic aberration. Very important. Now for submicroscopic microscopic area, medium to have high throughput and resolution required or a deeper look. Yes. So what we do, in our practice for day-to-day submicroscopic point mutation, deletion, time nucleoside. So we do a medium to high throughput. High throughput means आप डालो instrument machine में result fast आए. These are called high throughput. So these are all PCR machine which creates photocopy, which analyze the copy, and this is quantitative fluorescent PCR, clinical exome, sequencing, whole genome sequencing depends on positive predictive value. So this is the area where we take the DNA, we extract the DNA, and with this extraction, each and every DNA is small piece. So it is called library preparation. And with this library preparation, you need multiple number of extraction devices, very high cold centrifuge extraction. This is the area where we, with the qubit, we make the DNA, how much amount of the DNA is, whether we are doing the quantification of the DNA, because cell is having very small amount of DNA. We have to make DNA amplified so that with amplification, we can identify the defects in each and every pages and each and every letter. यदि आप मेरे को एक ही सैंपल दोगे डीएनए का, एक ही स्ट्रिंग दोगे, स्ट्रिंग घूम जाएगा. तो हम क्या करते हैं, इस स्ट्रिंग की कॉपी बना लेते हैं. अब इस स्ट्रिंग की कॉपी बनाने के लिए कॉपी कितनी बनी, कितने पिकोग्राम बने, दिस हेल्प्स. कॉपी कौन बनाता है? दीज आर ऑल पीसीआर मशीन्स. नाउ आफ्टर मेकिंग दिस डीएनए के जो छोटे छोटे, हमने डीएनए का पूरा कॉपी बनाया. अब इस डीएनए की लड़ी में मेरे को ईच एंड एवरी लेटर देखना है. तो मैं एक तरफ से पढ़ने जाऊंगा बिलियंस एंड बिलियंस ऑफ लेटर्स, उम्र बीत जाएगी. तो हम क्या करते हैं, जैसे नेचर क्रोमोसोम को छोटे छोटे स्ट्रिंग को छोटे छोटे पीस करके क्रोमोसोम बनाती है, हम हमारे डीएनए के लड़ी को बिलियंस ऑफ पीसेस में छोटे छोटे करते हैं. These are called library preparation, and each and every DNA piece will have some letters. Now this letter has to be read and compared with the normal letter. If it is comparing with the normal letter and it comes normal, then writing on this string is okay. But another comparison, if it is not comparing with, suppose this part or this part or this part, then we can identify where is the problem and what is the problem. Now, how to read this? We go for a machine which is called, we have to sequence everything. So this is called sequencer. If you take this sample is there, so there you can have machine is going on and it will give me the answer about comparing with the regular normal. If it is there, then we can identify the sample is normal or sample is problem. Is there is a problem in the globin gene? If there is a problem in the globin gene, then at what part of the globin gene it is missing? What type of mutation is there? These are so important to identify and do a prenatal diagnosis. And the thing is that with every chromosomal, with every disease, if we try to approach in a same manner, then the diagnosis will be more easy and effective, and we can give a very good answer to this. These are all very low temperature, minus 81. We store the DNA over here, and I think I have not made you too much loaded with the information or bored you. Thank you very much for joining me. And if I get a supposedly good response, I don't know how much I was, whether I can make you understand or not. Then, if I get a good response, then I will have again from my genetic lab the second part of the talk, how we do microarray, how we do exactly the libraries preparation, not going into the lab preparation, but more on clinical application. Thank you very much for joining me. Finish कहाँ से stop share किए get feedback. NVD.